In this video, we will dive deeper into the 4 guideposts that help explain how cancer likely develops in RUNX1-FPD, and how we might be able to stop it. Every RUNX1-FPD patient who has developed blood cancer acquired additional somatic mutations in their blood. Unlike germline mutations we are born with, the longer we live, the more somatic mutations we will have in our cells. The vast majority of these spontaneous mutations have no effect, but some do, and those are mutations that occur in genes that give cells a growth advantage or resistance to cell death, which, if left unchecked, will ultimately lead to cancer. This is why, as we age, our risk of cancer increases over time. When individuals without blood cancer have mutations in such cancer-associated genes in their blood, it is called clonal hematopoiesis of indeterminate potential, or CHIP for short. In the pool of stem cells in our bone marrow, each stem cell starts out without any CHIP mutations, the white cells on the far left. This example illustrates the process of cancer development over time in a person without RUNX1-FPD. They have no germline RUNX1 mutations to begin with. Over time, you can see how one cell all of a sudden gets mutation 1. Little by little, the cell with mutation 1 starts to take over the blood, crowding out the other blood cells without a mutation. This is the beginning of CHIP. And then mutation 2 happens, and now the cells with both mutation 1 and 2 start to take over. And patients may start to experience cytopenias, which are states where the body has lower than typical levels of red blood cells, white blood cells, or platelets, the last of which is called thrombocytopenia. This may be the most familiar cytopenia for this audience. At this point, the person still has what's considered CHIP. But eventually, over time, if another mutation, like mutation 3 happens, the blood system begins to fail and the cells with 3 mutations take over. At this point, the cells are likely considered malignant blood cells, meaning that they can no longer perform any of the normal functions of a blood stem cell. By the time the 4th mutation occurs, the cancer is robust, and the blood system is filled with many cells that can't make other blood cells, called blasts, and they keep multiplying and taking over. Individuals with CHIP have an increased risk of blood cancer, but not every person with CHIP will develop cancer. In the graph on the left, you can see that those with CHIP, the red line, have a higher probability of developing a blood cancer. What this illustrates is that we don't know which individuals will go on to develop cancer and which ones will remain otherwise healthy. In the group of individuals with CHIP on the right, we don't have any way of identifying the person who will eventually get blood cancer and which ones will be just fine. In order for cancer to develop in the body, cells receive multiple hits, or mutations. RUNX1-FPD patients are farther along in the potential path toward cancer because they begin with one of these hits at birth. For the general population, where each blood stem cell starts out with no CHIP mutations, cancer may occur by the 3rd or 4th somatic mutation. But for people with RUNX1-FPD, because they already have that initial germline RUNX1 mutation, additional somatic mutations can add up more quickly. Many already have thrombocytopenia even before getting their first somatic mutation, and the development of excess blasts, or malignant cells that grow uncontrollably, may already happen at the time of a second somatic mutation. CHIP is the first landmark on the road to blood cancer. Understanding CHIP will give us important information about the biological process required to go from a healthy blood system to a blood cancer. Knowing that CHIP occurs before blood cancer tells us that we need to delay or even stop the outgrowth of blood cells with somatic mutations in the blood of people with RUNX1-FPD. In one study from the University of Chicago, we learned that 67% of RUNX1-FPD patients below the age of 50 have CHIP. That is extremely high when we consider general population studies that show only 2 to 10% of non-RUNX1-FPD individuals above the age of 70 have CHIP. To wrap up Guidepost 1, since we know that in order for a person with RUNX1-FPD to progress to cancer, they must first develop additional somatic mutations, could we develop medicines that reduce the number of RUNX1-FPD patients who get CHIP and therefore reduce blood cancer risk? There are around 40 different genes that we know of that are considered blood cancer-associated genes. When they are detected in an individual, and it is determined that they have CHIP, researchers in the RUNX1 community asked, are there patterns of somatic mutations in these CHIP genes in RUNX1-FPD patients without blood cancer? If so, that could help us better understand how the cells start to grow out of control, and may reveal how to target them with medicines. Scientists have detected somatic mutations in RUNX1-FPD patients before they have any sign of blood cancer. In fact, researchers have discovered that there are some patterns. So far, it looks like mutations in blood cancer genes called BCOR, TET2, and DNMT3A are the most common. Remember that each stem cell starts out with a germline RUNX1 mutation. Those first few somatic mutations that occur are often in BCOR and TET2. Importantly, we also learned that in RUNX1-FPD patients with a blood cancer, the most common somatic mutation was in RUNX1 again, which means the other normal copy in RUNX1-FPD patients is mutated. And the next most common mutation was in a gene called PHF6. Somatic mutations in certain genes appear to occur just before cancer for RUNX1-FPD patients. So, we believe we may be able to target these genes with medicines to prevent them from taking over and developing cancer. For example, the mutations in the second RUNX1 allele and PHF6 are later events, meaning they happen either at a time of cancer initiation or after as part of the cancer becoming more aggressive. So there is a window of opportunity for prevention. If we want to prevent cancer, we need to eliminate, or at the very least control, the number of cells with mutations in genes like BCOR and TET2. Once mutations in RUNX1 or PHF6 occur, it's likely too late. Can we discover medicines that stop cells with BCOR and TET2 mutations from taking over? Can we target those cells specifically by killing them off and giving the cells with no mutations a chance to reclaim the blood system? These are the important questions that researchers are asking today. You may ask, why are the cells with certain mutations like BCOR outgrowing the other cells without somatic mutations? One potential reason is inflammation. Inflammation in the bone marrow, which is where blood stem cells live. Evidence of inflammation in RUNX1-FPD patients abounds. Special analyses of patient bone marrow prior to blood cancer and early preliminary findings from the natural history study run by the NIH suggests that inflammatory-related health issues such as reactive airway disease, eczema, allergies, and more appear common in RUNX1-FPD patients. The role of inflammation in cancer is undeniable. Inflammation, in particular chronic inflammation, results in an inflammatory response within tissues that is switched on for too long, causing cells to divide over and over again, increasing the chances for mutations. This can predispose to cancer and promote cancer growth. Current research suggests that the transformation to cancer requires both changes in the cell, like the somatic mutations we discussed, and the surrounding environment it lives in, their neighborhood. An increase in inflammation in the bone marrow, for example, can give a blood stem cell with CHIP mutations an advantage over healthy blood stem cells without mutations. What do we mean by advantage? Well, it's similar to how evolutionary adaptation works in nature, which is based on the theory of natural selection. For example, giraffes with long necks survived longer because they could feed themselves with the leaves from higher branches. The shorter giraffes can't reach the leaves in the large tall tree, and the shorter bushes and shrubs are depleted of any leaves due to competition with other animals. Since the giraffes with long necks could reach the leaves in the tall trees, they survived longer and therefore could reproduce more. They were also, therefore, healthier and stronger, so were sought after and selected for via mating practices too. So in the end, long-necked giraffes could better adapt to their surroundings, giving them a greater advantage in the African landscape. When it comes to the landscape of our blood system, it's possible that inflammation is a key factor in driving cancer transformation because it creates a condition that is difficult for healthy blood stem cells to survive in over time, and allows the outgrowth of cells with mutations that can survive better in a high inflammatory condition. Several ongoing research projects are focused on unraveling the role of inflammation and determining whether drugs that control inflammation could be a treatment approach to cancer prevention in RUNX1-FPD. This final guidepost might be the most obvious research approach, and that is to find medicines that increase normal RUNX1 protein levels, or medicines that can mimic normal RUNX1 activity. Could this turn a RUNX1-FPD individual's risk of blood cancer down to the same levels as an individual without RUNX1-FPD? Remember, each cell in a RUNX1-FPD patient has one mutated copy of the RUNX1 gene, but the other copy is healthy. Since each RUNX1-FPD patient produces on average about half the normal RUNX1 protein the body needs for a healthy blood system, perhaps we can artificially double the normal RUNX1 protein in a RUNX1-FPD patient's body through medication. Could we develop medicines that raise the production of normal RUNX1 protein from the normal RUNX1 allele, or create a medicine that could mimic RUNX1 so that the medicine replicates the effects of normal RUNX1? The challenge is to avoid increasing the levels of the abnormal RUNX1 protein in individuals who have a germline mutation that results in production of abnormal RUNX1. In patients with a RUNX1 germline mutation that results in no protein production, this approach would be easier. We've just reviewed the 4 concepts guiding RUNX1-FPD research strategy today, in 2021. Remember that in parallel to investigating all of these potential pathways to cancer prevention, our research community has been hard at work in developing tools we can use to begin to effectively use these guideposts to develop and test medicines in the laboratory. While researchers have just begun to understand these 4 key elements as clues to help along their path toward treatment development, other researchers have been laying the foundation needed to test treatments to target these elements. The most crucial tools necessary for developing treatments are cell-based animal models that mimic human disease. Without such disease models, it's impossible to test the behavior of medicines and other types of treatments sufficiently to determine whether they may be safe and effective in humans. In fact, The FDA and other regulatory agencies across the world who are responsible for ensuring patient safety require that such studies have been done in these model systems before allowing clinical trials in humans to begin. The RUNX1-FPD research community has been working in parallel to reveal these research guideposts, while also developing the tools necessary to ensure the quickest movement toward cancer prevention interventions. Lastly, and most importantly in the race toward RUNX1-FPD understanding and treatment development, is the incredibly powerful patient data. In the end, data from patients will provide us with the most critical clues on how RUNX1-FPD evolves to blood cancer. There's great power in numbers, and the more patients willing to share data by participating in research studies, the faster we as a community can learn and find ways to beat cancer. By watching this video series and informing yourself of the ongoing research strategies in place to develop treatments for RUNX1-FPD, you have already taken a critical step towards advocating for yourself, your family, and our RUNX1 community. Only together can we delay, block, or eliminate cancer.